Hello, welcome to the AP Biology live stream, units 1 through 4. I'm so excited that you're here. We are 2 weeks away from Cinco de Mayo. I know it's a really corny phrase, but I cannot for the life of me remember the last time AP Bio was day 1, 8 a.m. I mean, so soon, so soon. We are going to review units 1 through 4 tonight. Really quickly. So units 1 through 4 covering content, covering things that you absolutely don't have to know, which at this stage of the game is really good. Like if you can cross things off the list and say, I can ignore that. We're also going to be doing practice questions. So make sure that you have those ready for going. I need you guys to download the free study guide. So the free study guide is going to be located inside the ultimate review packet. I'm going to make this a little bigger so that you can see the QR code. Give it a scan. It's going to pop you right over to the ultimate review packet. And I'm going to show you where to go in order to download this document. It is multiple pages, but the reason that you should have it is it goes along with our presentation tonight. And this is going to make sure that you're getting the most active practice possible, taking some notes, jotting down key ideas so that you know how and what to focus on for different components. So popping over to the ultimate review packet really quick, this is where I need you to go. If you don't have an account yet, just log in with your Google address. It's free to make an account, and you're going to access a ton of free preview resources that I'll reference tonight as well. Once you're inside the packet, you're going to scroll all the way past the unit reviews. All these unit reviews have, study guides, summary videos, practice questions, practice sheets, practice videos, all 8 units. You see your practice exam down here, and you're going to go to live exam review. And since we are in the very first one, you're going to click on 1 through 4 guided notes. And this is going to have a link to this live stream just in case you happen to pop off and not be able to finish. It's going to break down all of the presentation parts, learning objectives, things you don't have to know. So the left-hand side here will have the presentation, and the right-hand side will have spots for you to put guided notes. The one thing that's not in here are those practice questions. So when we go through those inside of the exam simulator, you're going to want to make sure to jot down some of those key ideas. When you have these key ideas jotted down, that'll help inform your studying on the other side. So again, I'm going to let you guys come on, give you a couple minutes to get this document before we get started, and then we will go right back over to our review. So I'm going to leave the QR code over the next couple of slides as we go through units 1 through 4. Starting with chemistry of life. Now, this has such a small impact on the exam that this is, I don't want to say you can ignore it, but I will say that if it's giving you some anxiety, we're talking 8 questions, 9 maximum on unit 1 that you're going to see in multiple choice. So it's a really small portion of the exam. We're going to move on to cell structure and function, which is going to be a little bit more comfortable, especially if you've taken bio before. After that, we hit like the first really hard unit of AP Bio, cellular energetics. And that's because of cellular respiration, photosynthesis. They get a bad rap. Everyone gets their antenna up about them. There's so many terms and diagrams. We'll walk through that tonight as well. And then we're going to end with cellular communication, which is also a pretty heavy hitter on the exam. But before we get started, I just want to know, where is everybody from? So if you look in the back, mirrored, right? The back of my YouTube screen here, you'll see a little map of Philadelphia. So that is where we are located in Philly. And we are pretty far reaching, whether it's through a resource that you found through the podcast or the URP or the Exam Slayer. Where in the world are you coming from? A lot of the international students tend to use our resources when they're self-studying. So I tend to hear that as well. We are going to be about an hour long tonight. So that means about 20 minutes per unit. And that's going to include the practice questions as well. So we see Maryland, Connecticut, California. That's amazing, especially with different time zones. So thank you over in Cali. I know it's a little earlier out there. We're 9 o'clock on the East Coast right now. All right. Depending upon where you found us, maybe you found us through the original resource, the podcast that came out in 2020. YouTube, which started about 3 years ago. And then obviously the Ultimate Review Packet was about 2 years ago. And very recently, just this month, we have the Ultimate Exam Slayer. So layers on layers on layers of resources for you. This is going to be a saved live stream. One thing that won't save is the chat. So if you want to participate in the chat, make sure that you're a subscriber and make sure that you are active during the conversation tonight. Outside of that, this will be available for you to view up until the exam, which is in 2 weeks. All right, so as we say on our podcast, as we say in our videos, the first thing we have to do is zoom in to get some focus. Unit 1, chemistry of life. I don't think we can zoom in any farther. We're talking molecules. We're all the way inside of our cells. We need to get incredibly focused with those macromolecules and how structure influences function. These are the learning objectives. I am absolutely not going to read them to you, but they are directly from the CED. If you're not familiar with the CED, it's the course exam description, and it's what the College Board produces with every single iteration of their AP exams. And they revise them year after year. It's not a secret. I have it linked in the URP. You can find it with a quick Google. You want to make sure that you reference it at various times because it's going to give you exactly what you need when you need it. This means that it kind of removes the veil. Now, a lot of the questions that I get asked are, if I study, specifically just X, if I just use certain resources, will that be enough, right? If I just use your resources and I go over to, to AP Classroom, will that be enough? And it's hard to say because every student has different needs and different resources and really, different study styles. And that's going to influence how you should go about these last 2 weeks of studying. So will it be enough? I think it depends. There's like the surface level of knowledge, and then there's analyzing and applying that knowledge. And that's kind of where the AP aspect of this course comes into play. So it's not as much just knowing bio, but it's knowing what to do with the bio once you understand it. And so that's where we're going to focus some of our content today. All right. In Unit 1, don't waste your time learning specific molecular structures of any of the nucleotides, any of the amino acids or polysaccharides or lipids. So you might at this stage recognize, what adenosine looks like, but you don't have to do that on the exam. Things are going to be labeled for you. There'll be analysis questions that you could pull out from that. Let's start with water. Everybody's favorite solvent, and why it's so fantastic. It's because it's polar. So polar covalent bond within a water molecule is going to be due to the fact that the oxygen has electronegativity. And if you look in the red, larger atom of a water molecule in this drawing, you can see those extra electrons on the electronegative side. So water is not charged. Water itself has an uneven distribution of charge, and that's going to allow it to be attracted to other charged or polar molecules. So whether it's an ion or something else that's polar, it's going to have that attraction forming hydrogen bonds. So hydrogen bonding is not within a water molecule, even though hydrogen is present. That's not where we see that bond. We see hydrogen bonding between hydrogen and something else, right? Something else that has an electronegativity for that bond to form. And it's a weak attraction is what we see happening here. Because water has this polar covalent structure, it's going to give it a lot of really unique properties. We see cohesion, adhesion, that surface tension that allows it to have, the water bug feature where it can support life on the surface. It's also going to resist temperature changes. The expression of a watched pot never boils. Well, it's not true, but you have to get enough joules of energy within that water to break the bonds of hydrogen on that surface. So it does have resiliency, we'll say. All right, let's talk carbs. Everybody's favorite thing to eat. We see in an elemental ratio 1 to 2 to 1. That's your carbs, hydrogen to oxygen. So your classic C6H12O6, that's going to be what you see for your glucose. Now, you don't follow that exact ratio when you start to build polysaccharides because you have glycosidic bonds forming, right? So you're going to be losing some of those hydrogens in order to have those bonds forming. Glucose and is the one who gets all the fame, but we can see carbohydrates used in lots of different ways, structural as one of those components as well that we see inside cell walls. And different cell walls are going to use different structural molecules. So you're going to recognize glucose in many ways. You can find carbohydrates in ring structures, branch structures. They can be involved in cellular communication as well. And we'll see that inside of our lipid structure in just a second. All right, let's talk about proteins. When in doubt, a protein does it. Proteins have so many functions. I could never have listed them all here. Now, the reason you see the element sulfur in parentheses is not because you don't find them in proteins, because you find them in very, very few. Specifically, you're going to find sulfur inside of an R group. So when you look at the monomer of a protein, it's going to have an identical structure on all three sides: carboxyl, hydrogen, and an amino group. That fourth side, the variable, that's going to be your R group, and that can change from one amino acid to another. The chemistry of that R group is going to influence how that molecule, and if that molecule is attracted to or bonds to something else or is repelled. That will then influence its folding, and its folding will influence its function. So the way that this connects to later units has a lot to do with, is there an error in amino acid? Has an amino acid been swapped out so that you have a different structural component there? Because if you can't fold your protein correctly, then the function is not going to be the same. And that's going to have a big domino effect for the cell, the tissue, the organ, whatever that happens to be on down the line. So definitely something to be mindful of when it comes down to proteins. All right, lipids. The famous lipid, phospholipid, which again is why P is in parentheses. Now, if you were just to look at a carbohydrate chain, you might not instinctually know whether you had a fatty acid, which is a lipid, or whether you had a carbohydrate. But the ratio of those elements is what you need to pay attention to. In a lipid, you're going to see this have a greater ratio, 2-to-1, of your hydrogen to your oxygen. Now, as a whole, lipids are nonpolar, but you do have to recall that that's not really true for phospholipids. So phospholipids have a polar head and a nonpolar tail. So we refer to this molecule as amphipathic, meaning that different sides of the lipid have different attractions. Because of that, we get our bilayer and we have our membranes being formed, and that's going to form such a fantastic selective barrier for lots of different types of tissues. So looking here at what our phospholipids can be used for outside of what our lipids can be used for outside of the cell membrane structure, we're going to see things like storage. You're going to find things like in steroids, it can be hormone signaling. It can allow for different components of the fatty acid chains within the membrane structure, and we'll see that in just a second as well. All right, lastly here, nucleic acids. So looking at the nucleotide monomer of a nucleic acid, you find the classic, I'll call it, house, garage, sun, right? So the house would be your pentose sugar. You're going to have the garage, that's your nitrogenous base, and the sun is your phosphate group. Now, phosphate is charged. You see that minus sign. And so that's going to influence a lot of things about the way we have movement of our DNA molecule. We can see that again later on in gel electrophoresis as our molecules migrate inside of the chamber. When it comes to identifying parts of a nucleic acid, we want to be mindful of the fact that my pentose ring has the directionality. So referring to 1', 2', 3', and so on is going to tell you which orientation the molecule is in. Now, DNA and RNA, they get a large refer back later on down the line. So we're only going to briefly mention them here. As we get a little bit farther on, they're going to come back into play in much greater quantity. All right, unit 1, all the way through, and we're 15 minutes in. We have 4 of them to go. So we're about a quarter of the way done. But what I want to show you guys is some practice questions. And so what we're going to do is I'm going to switch over here to the Ultimate Exam Slayer, specifically the AP Simulator software. So if you have not yet seen this, I'm going to show you some of these features. I've had this open for 8 minutes, which is why my timer at the top looks so large. On the actual AP exam, it's going to count down, not up, but this is for practice. So you'll notice this looks super familiar, but it's not endorsed by the College Board. It's not created by Blue Book, that I can promise you. Couple key features here to note is that we can actually use like an elimination process here. So if we wanted to strike out options, we can do that. If we weren't really sure, we can mark the question for review, and I would see, oh, I'm going to have to come back to question 1 later on. The cool thing about this platform is that you can actually show the answer if you're not sure. And so you can either take the whole test or you can, go back to it later. You can take this multiple times. All of that would be really helpful for you. So I see a pretty good question here, really applicable to what we're working on right now. How many minutes should we spend per multiple choice question? So the real answer is 90 seconds, so 1.5 minutes. The answer I'd prefer you to use is about 1 minute if the question doesn't have a stimulus or a prompt. So the example we have on the screen right now doesn't have a particular prompt. So this might be a faster question. I'm going to show you one later on that will have a stimulus. So it takes longer to read. So yeah, if you guys happen to know the answer to this question, it's been on the screen for a couple minutes, please do answer in the chat. I'm going to talk through it a little bit and then show you how the software works. And we're going to come back to this a couple of times. So we're going to use this exam simulator software for 2 questions in every unit tonight so that you guys can see how it works. So if we're looking at this particular question, we can see that, Rosalind Franklin's diffraction images, super famous, photo 51, and every single option here, like, it's true about DNA. So it's a little difficult to maybe narrow this down a little bit. If I look here at these choices, and these, I should note, these are all released AP questions. So these are not written by me. You can access AP style questions written by me in the Ultimate Exam Slayer, and I'll show you that a little bit later on as well. But this software looks exactly the same as what you'll find in Exam Slayer. So if we look at our choices here, my ratios of base pairs are constant. I see that looks familiar to me. I know my strands are anti-parallel, but I'm going to cross this off as a choice because it has nothing to do with Rosalind Franklin. And I don't think here that being anti-parallel has anything to do with this question. So at this stage, I'm going to show answer so that you guys can see how this program works. And I can see that D is the correct answer. Photo 51, it had that X structure here. Watson and Crick, they confirmed the structure and assumed anti-parallel. Option A here, that's because of Chargaff's rules. That might look familiar to you. So I'm not going to read through this whole section, but know that every question that you use with Exam Slayer and with this simulator has this particular feature in mind. Let's go on to the next question for Unit 1. So we're looking at a protein suspended in water, and it's reacting to that water. So it's folding here. When it folds, it's going to have non-polar R groups is going to be kind of like huddled in the middle. And things that are polar are going to be in the surface of the protein. So remember, polar refers to an uneven distribution of charge. And so if I'm looking here and I'm trying to determine why is it folding in this way, we can have a couple different options here. So if you do know, pop it into the chat, and we can go over these questions as well. So looking at my choices, ionic bonds with the charged ends of the water. That doesn't look too promising, does it? Non-polar R groups, bulky doesn't seem to make sense to me as we go, because although R groups can have different sizes, that shouldn't have any influence here as to how it's bonding. So I'm going to go here and hit show answer and see that the correct answer here was C. Non-polar R groups cannot form hydrogen bonds with water, and so they're going to essentially be pushed away by the water. If I go down to my explanation, I can see a little bit more information as to why those other options are incorrect, and it does have to do primarily with hydrogen bonding. This did seem to be describing an integral protein within the software, but without an actual accompanying diagram to see that, we may not know. All right. We will come back to it. I promise. That's Unit 1. All the way start to finish. If you feel like you need more help with Unit 1, everything about Unit 1 inside the Ultimate Review packet is free preview. So you can get summary videos, study guides, all the answer keys, practice sheet, the answer key practice sheet. And I actually talk through and walk through all of those practice questions in video format as well. Plus, there's 10 Unit 1 multiple choice questions inside there. So what you see in Unit 1 is actually mirrored inside everything else for all 8 other units. So you can get an idea of what you like to see as far as the process of the packet go. All right, let's keep going here and start Unit 2. So one of my favorite quotes about Unit 2 is that there's no elephant-sized amoebas. If you don't know, an amoeba is a protist. It's actually the symbol on our podcast is an amoeba in terms of how it's described. So the question really here is like, why are there no elephant-sized amoebas as we go? If you are just joining us, I do want to make note of the fact that we're going through guided notes that you can absolutely download from the Ultimate Review packet. It's totally free. We've already done Unit 1, but all of this is saved. So if you've missed Unit 1 and you're just starting here for Unit 2, you can still catch up with that content later on. All right. Absolutely not going through learning objectives for Unit 2. There are so many. They're all in your guided notes, so go check them out. Things you don't need to know. All right. How lipids are synthesized, specific ways that these enzymes are packaged inside of vesicles, the specific role of the Golgi in that phospholipid process. So one of the things that students unfortunately tend to fall in the trap for Unit 2 is like staying in, I'll call it baby bioland, meaning it's not necessarily AP level. There are things that you may have learned in bio or even started this unit off with like making a lot of analogies to a city or a house to what those particular organelles do. Just be mindful that those types of answers on the AP exam, they're not going to earn you any points. So you want to be as specific as possible to the applied function within the question that as it's being asked. So just be mindful of that as you go forward. Anything with an asterisk on this list comes back in later units. So if I were you, those would be the particular questions that I would focus on. One quick question here I want to address. So how can we shore up on the info that we just don't know from questions we struggled with? That's a great question. I would keep a running list and focus my energy only on those things. So when we're done doing the multiple choice inside the exam simulator, it's actually one of the ways that you can direct your functioning. because it's going to show you which units and specifically which topics you didn't feel confident in, and you can go and direct your attention for that as you study in these last 2 weeks. All right, surface area to volume ratio. One of my favorite topics. It's kind of always the answer. So as you get too large, you run out of surface to move things across, whether you need to take in a nutrient or get rid of a waste. You kind of struggle to stay alive. And so you have to kind of find this Goldilocks zone. The way to do this is a lot of folding. See, that's why the mitochondria, inner membrane, the cristae is so heavily folded. We see folds inside of the small intestine. I think the analogy in the small intestine is it's something like if you were to take your intestines and flatten them, you get like half the surface area of a tennis court. It's a lot of surface, which is really necessary in the intestines because of, absorption after digestion. So you want to make sure that you can do that. If you struggle with these types of questions, I have a lot of practice inside the URP. I also put an entire, I should have put this in here, but an entire math review worksheet inside of the Ultimate Exams layer. So if you feel like math is a weakness for you, you definitely want to go check that out. All right, let's look at our friend here, phospholipid bilayer. So blue, phospholipid head, polar. Black 2 lines, fatty acid, nonpolar tails. That's going to compose your bilayer. Purple, in the middle, we have a long integral protein. This looks like a channel protein, doesn't seem to be gated. In the green, we have some carbohydrate chains. Now, if it's attached to a lipid, that would be a glycolipid attached to a purple protein here. glycoprotein. The yellow would be a steroid. All of this, I don't think you'd ever be asked to label. However, knowing how these different components impact phospholipid structure function, specifically with regulating what goes in and what comes out, that's where I would put my focus. And this all leads down to, how do things move across the membrane? So we have two types of transport here, active and passive. And that's going to depend on which direction are molecules moving and how is the energy being used, and if the energy is being used. So when I say energy, I'm specifically talking about ATP, because obviously some type of energy has to be moving molecules in passive transport, and that's kinetic energy. The molecules they're bumping into each other because they are always in motion. So if I'm on the passive side, I can either move directly through the membrane. Osmosis would be water. Diffusion is everything else that's small and nonpolar. Or if I'm a little larger or charged, but I'm still moving down my concentration gradient, I'm going to move by facilitated diffusion. That's going to involve a protein. On the active side, can also move through a protein. These are going to be things that are more like pumps, like the sodium-potassium pump, or a proton pump like we see inside of the electron transport chain. Those will all be useful for that. You can also move through vesicles, so endo and exocytosis. And we're going to see one of those in a practice question in just a second. So I like this kind of overview perspective on transport, but we're going to need to dive a little deeper when we do application of these questions. I'll make this a little bigger for you to see. All right, tenacity and water potential is definitely something students tend to be a little afraid of. Don't be afraid. Tenacity. We are specifically describing solute concentration and whether it is greater or less than our water concentration. So when we talk about a hypertonic solution, a lot of solutes, less water. But this is concentration. It's not amount. And I'm blocking that part where it says that. So I'm going to take my banner down so you can see this a little bit better. There we go. That's so much better. When I am hypotonic, I have a lower solute concentration. These are comparative terms, so we need to make sure that we are always comparing them to something else. This could be extracellular to intracellular environment. It could be an artificial cell. It could be a U-tube, like not Y-O-U, but U-tube, in that we are going to see water volumes change. And then we have isotonic, and that's no net water movement. And what's important about that one is that water is always moving. Let's just be clear about that. It's just not moving in any particular direction. We also have to be mindful of solute movements, because solutes can also follow their own concentration gradient. But concentration and equal concentration does not mean equal amount. This is why we end up with cells that go through lysis. This is why we end up with cells that shrivel or crenate. This is how we end up with cells that go through plasmolysis if you're on the plant cell side. So all of these specific terms and conditions, those all are impacted by how much water do you have, where the solute's going as we go. Can't water, good question. Can't water do active and passive transport? So it is passive, but you are referring to an aquaporin. So water is polar, but it's also super small. So we can move by osmosis, high to low, directly through the phospholipid barrier, kind of like sneaks through because it's so small. But if it has to move in larger quantities, that's when it uses a protein channel. And the one that's specific to water is called an aquaporin. So great question. All right. If you're a little wonky with water potential practice, I do have, as part of the Unit 2 practice sheet in the URP, whole bunch of questions. And one of them specifically is like an FRQ style practice question for water potential. It also includes a video explaining this and other things inside that practice sheet. So if this is one of your weaknesses, definitely pop over and grab that and get a little bit of extra practice. All right, 9:30, we are on time. Let's look at two more practice questions inside of the AP exam simulator. And this is going to lead us to This is going to lead us to the next stage. So moving into question 3. I'm going to try to make this a little bigger for you. Let me see if I could do that. There we go. All right. A pathogenic bacterium has been engulfed by a phagocytotic cell as part of a nonspecific innate immune response. Which of the following best illustrates this response? And in your head, you're going, I don't know anything about the immune system. It's not something I was required to know. And that is very correct. However, phagocytosis is a process you are expected to know as active transport with a vesicle. So it's non-specific, and we're looking for something that's going to get rid of a bacteria for us. Thinking back to our organelles, we want to make sure that we have some mindfulness of what the organelle functions are, because that's really where our answer is going to be. Now, again, we're in the in Exam Slayer software, so crossing out is an option for us. I'm going to turn that on so that we can cross out some choices if we want to. All right, the first one has a mitochondria in it. I don't like that option. Mitochondria don't do things with bacteria. We should be in cellular respiration land for that. All right, B looks promising. I've heard of an antibody before. I haven't studied it. I don't know where they came from in this picture, but it looks like they're trying to attack this in some way. I don't know if that's non-specific, but I'm going to leave it for now. Then I look at option C. I see a lysosome. I like that choice. And mitochondria coming back for D. I don't really like that one either. So let's click show answer, make this smaller again. And again, it will go through the responses for us. This is, again, an approved question, 2012, CED is what this came from. And we're looking at the option here, which includes a lysosome, is the correct answer. That's where we want to spend our focus on. All right, let's move on to the next question. Make this a little bigger for you. If we have ATP breakdown hydrolysis is inhibited, which of the following types of movement across cell membranes is also inhibited? Okay, if we can't hydrolyze, meaning take off a phosphate group from ATP, what can we also not do? So we're looking for something that would be descriptive of active transport. Active transport. So a couple keywords that you should be looking for, things like against, that would imply concentration gradient, things like low to high, things like into something that's more crowded. So let's see if there any of the options here have that in it. And I do see some options here. So C has the word against in it. That to me is a clue that it's probably C. Let's see if it is and hit show answer. Alicia Hill coming up hard with C as we go. Let's see if she is correct. Show answer. And she is. Look at that. Going down to why the other options are not correct. Oxygen, super small, nonpolar. That needs simple diffusion. That's not going to be a situation. There's aquaporins. That's B. We already mentioned that. And then moving into my last one, facilitated diffusion. So facilitated just means made easier. So fácil, if you happen to study Espanol, is where that term is going to come from. And so we can use that for all of our associations in making diffusion a little bit easier with the use of a use of his I just saw a comment in the chat. The use of sorry, one of my students coming up hard in the chat for this. It's a student that I didn't even tell about the live stream, so that's rather impressive. All right, we're going to move on to Unit 3. Again, if you have not gotten the study guide for this, make sure to grab it from the Ultimate Review packet. It's in the live stream folder so that you can follow along with the presentation. Bio has to follow the laws of thermodynamics. That's absolutely true. Here are our learning objectives. It's going to be something that you can access inside the packet, so I'm not going to go through all of them. Unit 3 can be a little scary for students, but we will get through some of those harder things. Don't have to know from Unit 3. You do not have to know anything about calculations of pH or Gibbs free energy. You're going to see it inside of diagrams, but no calculations. any of those specific steps, molecules, enzymes of either cycle, specific carriers or intermediates in the electron transport chain, you don't. I'll be honest, if they want you to do something with an electron transport chain, right? The AP Bio they, with the electron transport chain or with the Calvin or the Krebs cycle, they're going to give you a diagram. They're at least going to give you like a little bit of a diagram for you to associate to that. So you don't have to really, one, never, ever memorize. But two, you don't have to panic when you see these types of diagrams. Let's start with enzymes though, because in reality, if you look at those learning objectives a little bit closer, enzymes are most of Unit 3. Like cellular respiration and photosynthesis, they're one line a piece. They're really, really small, and they fill up a lot of space, but they're not huge points of emphasis. So let's talk enzymes. Made of protein, so they have specific structure for a specific function. They're going to bend and fold into tertiary or quaternary structure depending upon what their active site needs to fit. The lock and key analogy, really outdated for enzymes, but it does have to have a slot, we'll call it, for our substrate to fit and bind. And that's going to assist in particular reactions. So the phrase that's used, unfortunately, too often is speeds up chemical reactions. Okay. What it really does is lowers the activation energy. It makes it so that the reaction can occur while using less energy to do so. And because of that, more reactions occur over time. It's not manipulating time, it's manipulating the energy required. So we see two graphs here. That y-axis gives free energy. And you can see that some reactions are going to be energy storing and some are going to be energy releasing. If we were to compare these two graphs from a photosynthesis cellular respiration perspective, you're going to have photosynthesis being an energy storing process or endergonic, and exergonic being an energy releasing process. But regardless, you can't make energy. You just transform it. Okay, so if it starts off as energy from the sun, we are going to then capture and transform and temporarily store it, well, several times, but eventually in a molecule of glucose. And then we need to use that energy for a different, for maybe motion or movement. Maybe it's active transport, or perhaps it's kinetic movement. We're going to then transfer the energy to something else. So not created or destroyed. Really important to be mindful of as we describe energy transformation. All right, ATP gets all the glory. ATP is it's not stored energy. It's one of our misconceptions we tend to use. It's just temporary usable energy. It's like going to an energy currency exchange, and you need to make it so that your cells can, understand how to spend it. And the way that this works is primarily through hydrolysis of that third phosphate group. Now, you can get more energy from ATP if you hydrolyze the second phosphate group. It takes a little bit more to do so. And so it's not as, we'll say, energy efficient to do that as a process. All right, photosynthesis coming in strong. Remember that you don't have to know all the steps. So if you have like a particular textbook that you've been following, you might have had more like 5 steps seen on one review guide. And oh, no, these are the 6 steps of photosynthesis. And it's like, oh, no, really, it's 7.5. None of that matters because the steps are only made so that you can understand it a little bit more easily. It's nothing that occurs in specific sequences and stops until the next step. So just kind of let that go if you've been stuck in step land. If you feel that that's useful to you because you like to learn things in chunked groups, totally fine. But don't get stuck up on it. Focus instead on what goes in, what comes out, where, and why. Like what's being done in that. So we're in the chloroplast. We are specifically in the thylakoid membranes. We're going to see an analogy here to the mitochondria because they're both endosymbionts and that they have double membranes, their own ribosomes, their own mitochondria, their own DNA, all these things. So this is what we need to be mindful of as we're kind of going through all of these. So let's look at something a little bit more scary, kind of big picture stuff. So in the ETC, excuse me, we are going to be splitting water, releasing oxygen, and forming our proton gradient. When you build the electrochemical gradient, we're going to have hydrogen ions moving across the membrane. That is going to allow for the formation of NADPH and ATP. The goal of the light reaction is to capture energy from the sun and transfer it into these temporary energy carriers. These temporary energy carriers will then go to the Calvin cycle. The Calvin cycle occurs in the fluid portion of this chloroplast. So that's going to be within the stroma. And it's going to fix carbon. So by fix carbons, that means it's going to take CO2 and stick it together in order to form a carbon compound. And that carbon compound will be where the energy is then transferred to. The Calvin cycle makes a 3-carbon sugar. So we have to turn the Calvin cycle twice to get glucose. And that's really important. All right, moving into cellular respiration. So same strategy, what goes in, what comes out, where does it happen, and why? When we go through different components here, we can see that although glycolysis is anaerobic, it can be included in the cellular respiration aerobic pathway. So I take my glucose molecule, I cleave it in half, I end up with 3-carbon pyruvate. That pyruvate will go on an adventure, either fermentation pathway or the into the mitochondria for cellular respiration, further breakdown and release of that energy. The fermentation pathway does not make ATP. It regenerates NAD+, so that glycolysis can occur again. It's going to be glycolysis that is going to produce the ATP when it's going to break down my glucose molecule. If we're going into the mitochondria, we're going to make significantly more ATP, and that's really where the bang for your buck comes from that perspective. So let's look at the next 2 phases, the Krebs cycle and the electron transport chain. We kind of have a mirror image of what we saw in photosynthesis. So if we were going to be storing in carbon compounds, here we are going to be releasing from carbon compounds. And so the goal of my Krebs cycle is to finish breaking down my carbon and making and transferring energy into NADH and FADH2. Those are electron carriers to drop off at the electron transport chain, and that's going to build my proton gradient, build up that ability to produce more ATP. All right. Moving over. If you find you're confusing them, and actually I see some comments in the chat, really confusing. Citric acid cycle, Krebs cycle, Calvin cycle, they have a lot of names. TCA cycle, definitely head over and go through some of the practice in order to get that a little bit straightened out. Okay, so you know the drill by now. Let's go through 2 practice questions for Unit 3. And because my voice is going, I'm going to give you guys a second to read this while I get a sip of water. All right. How nice to give us the chemical equation for photosynthesis. which is lovely. But here we're talking about, all right, let's make oxygen radioactive. What's going to happen if the oxygen in my water molecule right here is radioactive? Really what this question is asking is where does it end up? Like, how do we know what's going to be happening next? And so looking at my choices here, the red herring is absolutely here, meaning that they want you to pick the one that has CO2. But oxygen and CO2, they don't go together. So just be careful of that when we're going through our options. If you want to then use the the cross out option here, we can do that, but we're going to hit show answer because at this stage of the game, we are aware of how the program works. So just be aware of the fact that we want to not pick the ones that have really the Calvin cycle. It has nothing to do with water splitting. We also don't want to be the one that has to do with CO2 because CO2 and oxygen don't go together. We get the oxygen when we split the water molecule. All right. Next question. I like this one a lot because this is actually going to show you the split screen functionality that we see inside of the exams layer. Excuse me, as my voice goes, I'm going to make it bigger and get rid of my picture so you can see a little bit better. So this question that we see here is actually going to be really indicative of what you would more likely see on the AP exam. This is from the 2020 CED, which is the one that's currently being used. So if we hit here, show answer, we can see that we're going to denature a protein. Now, one of the trip falls of this question is denaturing a protein is going to be losing its 3D shape. So we're breaking really R group interactions here, some hydrogen bonding. We're not breaking peptide bonds. So nothing is being disassembled in primary structure. Really important to take note of. Last unit is going to be unit 4, and we're only running about 4 minutes behind, so I think that's rather respectable. Learning objectives, one more time, you can grab that inside of the guided notes inside the Ultimate Review Packet. If you haven't yet downloaded that, make sure to do so. Do not need to know specific cyclin CDK pairs or growth factors. When we look here at our signal transduction pathway, 3 steps: reception, transduction, and response. And I pulled this particular diagram on purpose because it looks scary, but it's not. It's just showing you different stages of this process. Now, the ligand is going to bond in different places depending upon its chemistry. So if it's going to be charged or large, that's extracellular. Nonpolar is intracellular. So in this particular FRQ, if you want to try it, you can get that off of AP Central. It's going to allow you to walk through kind of like the different phases here. Things to note is that things are both active and inactive, depending upon the stage of the transduction pathway and the type of response necessary. The response in this example is chloride ion secretion by the cell. Two types of feedback mechanisms to focus on, negative and positive. The overwhelming majority of feedback mechanisms are negative. Positive feedback loops, they're super temporary. They have a really short intention for something specific for the organism. So all the examples here, childbirth isn't happening during most of people's lives. Fruit ripening is a really, really short sequence of time. You should not be clotting your blood all the time either. So it's going to have a lot of different pieces, not an overwhelming component of that. Most things are negative feedback. And then one of the last pieces of Unit 4 is the cell cycle checkpoints. Students get stuck up on these a lot. What you want to put your energy into is what's supposed to happen at these steps. And so then the checkpoint, as a consequence of that, is going to say, well, if I didn't replicate my DNA in the S phase, that would be a big issue. If I don't have appropriate cytoplasmic size, that would be an issue. If I don't have chromatids attached to spindle fibers, that would be an issue. So these are going to be all your different checkpoints that you see throughout the cell cycle. Not shown on here is G0. So sometimes we'll see a non-dividing phase. A cell will exit the cell cycle during the middle of G1, never copy their DNA. That would be something like a nervous system cell. And lastly here, good old PMAT, prophase, metaphase, anaphase, telophase, and cytokinesis at the very end. So making it a little bigger for you. Remember that the goal of mitosis is to make an identical copy. So 2N to 2N. We're making one more copy of what we just had. This particular cell is shown 2N equals 4. So this is a 4 chromosome cell. Not pairing as homologous chromosomes because I'm in mitosis. So we're going to see sister chromatids and no crossing over because we're making an identical copy through all of this. All right. Before we get to our final recap, I want to make sure we get our last 2 questions in for practice. So I'm going to head back over to the practice questions and we'll do our final 2 in the packet. So number 7 here. If chemical signals in the cytoplasm control the progression of a cell to the M phase of the cell cycle, then fusion of the cell at G1 with a cell in early M phase would most likely result in which of the following? This is something that we've seen. You put 2 cells next to each other at different phases, they can influence each other. So the question here is, what influence would this cell have on that G1 cell? All right, had a little bit of time to think. Go here, we're going to hit show answer. And I can see that we're going to get condensation of the chromatin as if it were going to divide. And clearly that's going to be an issue. My G1 cell hasn't even made a copy of its DNA yet. So we're not going to want to see that coming through all the way. All right, coming back, we're going to do one more question. About the endocrine system. So much like the previous question where you're saying, I didn't have to study the immune system. Why would I need to know the endocrine system? But in reality, they can use, again, any application of biological concepts. They just have to provide you enough information in order to answer the question properly. So looking here, we're looking for an option that is going to designate negative feedback. And the one that's going to designate negative feedback is going to be returning to homeostasis, not removing farther away from homeostasis. So clicking my answer here, that's going to be the blood glucose levels. And I saw a question earlier, what feedback mechanisms do humans have? The answer here is that humans have both types of feedback mechanisms, because they have to have circumstances in which they go farther from homeostasis and those in which they come back to homeostasis. So we're going to see that in both aspects, not just in one. So looking at these other options here and just why they're not correct. The last choice D, that was describing gene regulation. So that's not going to be anything that we're going to see. So because I've come to the end, I'm going to hit finish test and hit submit. Now, hopefully, in this context, we actually didn't answer any of the questions. I don't know if you actually noticed that. But this is going to show us, you know what, you did flag a question, and this is the way the breakdown would go. I'm not getting a score because, again, I didn't actually answer any of the questions as we went through them. We were just looking through with the answers. Had you clicked and answered the questions, it would have given you a proper feedback on the other side, which we did not do. So to recap all of my parts here and making sure that we have the 4 different units, we have chemistry of life, cell structure and function, cellular energetics, cell communication, and cell cycle, with some really, really key features in each of them. Again, this is on your download. If you're taking other AP exams and you want to try out this software for yourself, we have authored, the URP team has authored 10 questions that you can try for free in all of the subjects. So this QR code is going to lead you to the Ultimate Exam Slayer free preview for all of those different subjects. If you are interested in trying out this AP simulator software for yourself, again, this is also on your packet. So with that, we have another live stream in 5 through 8. It's going to be on the 25th. And we will, again, go through practice questions. We'll go through content. There'll be another packet for you to download if that is going to be something that you're interested in. I do want to say thank you for coming. I appreciate you participating in the chat. I want to see if there's any other questions that I can help with that I haven't seen go here. All right, the format. Okay, MCQ format hasn't changed much. But the FRQ has changed just this year. So that's going to have questions embedded in that. And we'll actually go over that when we do the practice test on the 29th. In regards to the multiple choice structure, you are still going to see those that are clumped together with problem sets and those that are individual. Because multiple choice is fully digital, it'll have that split screen like we had with the Tripson question, where there's be a stimulus or a graph or a chart on the one side, question on the other. And then the questions will update as it goes through with the stimulus. On the FRQ, you're going to still be handwriting those. So all of the questions are viewed digitally, but it's not going to be separated out. So with that, I do want to say thank you for coming. Make sure that you pop back over and you check out the next live. We only have 2 weeks left. So with only 2 weeks, it's going to be a real crunch time. Being the first exam, it definitely gives us, I think, a little bit of an advantage because we aren't yet studying for all of the other ones yet. I know most of you probably take like 5-6 APs. So I do appreciate you coming tonight. Best of luck on the exam if I don't see you at another live stream. But otherwise, I'll see you next recap.